You are a hungry Pleistocene gatherer staring at a bush of vibrant red berries. There is no Google, no poison control hotline, no expiration dates to check. You are starving. Your tribe is watching. Eat the wrong leaf, and your nervous system violently shuts down. Eat the right one, and you survive Tuesday. So, who takes the first bite? The plant kingdom is not a peaceful garden offering up snacks for passing bipeds. It is an active, merciless biological warfare zone. Animals can run, hide, or bite back when threatened. Plants are rooted to the ground. They cannot flee. So, to avoid being eaten into extinction, they became master biochemists. Over hundreds of millions of years, vegetation evolved a spectacular arsenal of neurotoxins, cardiac glycosides, and vicious alkaloids designed to do incredibly unfun things to the biology of anything foolish enough to take a bite. Some compounds paralyze your diaphragm. Some disintegrate your liver. Some just make you violently evacuate every fluid in your body until you expire from severe dehydration. Nature is beautiful. So when an early hominid band wandered into an entirely new valley, they were essentially walking through a terrestrial minefield of chemical traps. Every new root, seed, and leaf was a massive gamble with death. Our ancestors did not have the luxury of a botanical field guide. They had to figure out the menu from scratch, using nothing but their senses, their raw intelligence, and a grim willingness to sacrifice a few unlucky individuals for the greater good of the gene pool. The first line of defense was entirely hardwired into our biology. Evolution gave us a built-in poison detector: our tongues. If you have ever wondered why toddlers instinctively gag at the sight of Brussels sprouts, or why black coffee requires such a forced, acquired taste, you are witnessing an ancient survival mechanism in action. Most deadly botanical compounds, particularly toxic alkaloids like strychnine, quinine, or atropine, taste incredibly, overwhelmingly bitter. Over millions of years of primate evolution, our ancestors developed an extensive family of taste receptor genes, specifically the TAS2R family. These receptors are literally designed to sound a massive physiological alarm the second a bitter molecule touches your palate. The brain registers the flavor and immediately screams at the body to spit it out. This innate aversion saved countless early lives. If a curious forager bit into a lethal nightshade leaf, the intense, astringent bitterness would force them to eject it before a lethal dose could hit their bloodstream. But this system was not flawless, not even close. Some deadly plants taste perfectly fine. The deathcap mushroom, famously, is said to be quite delicious and savory, right up until your internal organs slowly liquidate over a period of 3 days. When our chemical senses failed, we outsourced the problem to the locals. Ancient humans were apex observers. They spent their entire lives reading the intricate rhythms of their ecosystems. If they encountered a strange new fruit, the easiest way to test its edibility was to see if anything else was actively eating it. If the local macaques or foraging bears were gorging themselves on a particular patch of wild figs and not dropping dead out of the canopy, it was a fairly safe bet that the apes carrying the spears could eat them too. This practice, rooted in basic zoo pharmacognosy, provided a massive shortcut for prehistoric survival. But copying the wildlife was an extraordinarily dangerous game. Biology is highly specific. What is a nutritious afternoon snack for an avian digestive tract might be a fast track to the afterlife for a primate. Birds, for instance, are completely immune to capsaicin, the volatile chemical that makes chili peppers spicy. Many rodents can comfortably digest berries loaded with compounds that would send a human heart into immediate cardiac arrest. Watching a squirrel survive a meal was a decent starting point, but it was never a guarantee. Eventually, observation and biological instinct had to give way to raw, brutal experimentation. This is where the absolute origins of the scientific method truly began. It was not born in a sterile, brightly lit laboratory. It was born in the dirt, driven by sheer caloric desperation. When faced with starvation, someone had to step up and be the prehistoric guinea pig. Anthropologists strongly suspect early humans utilized a primitive, highly intuitive version of the universal edibility test. It was a methodical, painfully slow process of escalating biological exposure. First, you take the unknown plant and rub it against the sensitive skin of your forearm. Wait a few hours. Any rashes? No? Good. Next, you rub the sap on your lips. Wait again. Any burning, numbness, or aggressive swelling? No. Great. Now, chew a tiny piece and immediately spit it out. If your throat does not instantly close up, you swallow a microscopic fraction. Then, you sit by the campfire and wait to see if you start hallucinating ancient demons or foaming at the mouth. Usually, this incremental dosing allowed a desperate forager to identify a poison and walk away with nothing worse than a terrible stomachache and a lingering sense of regret. But sometimes they miscalculated. Sometimes the neurotoxin was simply too fast, or the experimental dose was just slightly too high. Someone died. It was tragic. But more importantly, it was data. That macabre data formed the bedrock foundation of human dominance. Because our ancestors did not just passively memorize what killed them, they actively sought ways to hack the very plants that desperately wanted them dead. This is where early humanity transitioned from mere surviving apes to undisputed masters of their environment. Through astonishing mechanical ingenuity, they figured out how to forcefully deactivate biological weapons. Take the common acorn. Packed heavily with complex tannins, raw acorns are severely bitter, highly toxic, and will entirely wreck a human digestive tract. But ancient hunter-gatherers discovered that if you systematically crush the acorns into a fine meal and repeatedly run fresh water through the paste, the water binds perfectly to the water-soluble tannins and leaches them completely away. Suddenly, a toxic tree nut becomes a highly nutritious, life-saving staple flower. Or consider the cassava root. In its raw, natural form, cassava roots are heavily loaded with cyanogenic glycosides. If you aggressively chew and swallow a raw cassava root, your digestive enzymes immediately convert those defensive compounds directly into hydrogen cyanide gas. Instant death. Yet, indigenous peoples in the Amazon basin figured out that by intensely grating, fermenting, pressing, and extensively boiling the root, the cyanide gas safely evaporates entirely into the surrounding air. They turned an underground death sentence into one of the most vital carbohydrate sources in human history. They didn't just survive the chemical warfare of the plant kingdom, they actively dismantled the biological bombs and ate them for dinner. This encyclopedia of survival wasn't written down. It was passed through the most powerful technology early humans possessed: cultural transmission. Stories, myths, and taboos encoded the botanical rules of life and death, transferring hard-won data across generations. Every time you stroll through the produce section of a modern supermarket, you are standing on the shoulders of millions of years of brutal evolutionary trial and error. You are alive right now because countless forgotten ancestors looked at a strange berry, took a terrifying bite, and either lived to tell the tale or became a stark, fatal lesson for the rest of the tribe. Their bravery built the modern diet, but their evolutionary ghosts still haunt your DNA, warning you every time you sip a bitter cup of coffee.